Welcome everyone to the March 25 International Lecture on Chromosomal Instability, organized by Florian Markovits from the Cambridge Cancer Institute, Orly Ernst from BKFZ, and Heidelberg, and myself, Isidro Cortez from the EMBL-EBI in Cambridge, UK. This year we have an amazing lineup of speakers, as you can see on the screen, so pencil in the dates in your calendars, please. And remember that we will put all the lectures in YouTube as well in the channel that's indicated on the screen. Today, it is my great pleasure to welcome Dr. Sam Bakhoum, Chief Scientific Officer, co-founder, and board member of Volastra Therapeutics, a New York-based biotech company focused on the design of novel anti-cancer therapies targeting chromosomal instability. Prior to joining Volastra full-time, Dr. Bakhoum was an associate member in the Human Oncology and Pathogenesis program at the Memorial Sloan Kettering Cancer Center in New York City, where he also served as a practicing radiation oncologist. Over the years, Sam has made really key contributions that have dramatically advanced our understanding of the role of chromosomal instability during cancer evolution, progression, including metastases. And on top of that, he's a great speaker, and I'm most happy and honored to have you in this lecture series, and I very much look forward to your talk. So over to you, Sam. Thank you.
Thank you very much, Isidro, for this very kind introduction, and to all the organizers for inviting me. And I just want to draw your attention to this amazing lecture series, and here's a poster for the talks to come as well, so make sure you put them on your calendar. So as Isidro mentioned, I recently took over the role as Chief Scientific Officer, and I'm a co-founder of Volastra Therapeutics, which is a company, a biotech company, clinical stage company that aims to target chromosomal instability for the treatment of cancer. And today, the theme of my talk will be kind of a series of 3 stories that basically summarize a few years of research on our quest to understand how chromosomal instability, or CIN or CIN, drives cancer progression, promotes cancer immune evasion. These are my disclosures, most of which I've already mentioned. So I don't need to spend a lot of time introducing the concept of chromosomal instability to this audience, sorry. But I think it's still worth just looking at karyotypes once in a while, because often in this day and age, we often look at next-gen sequencing and genomic data, which obviously is extremely informative. But it's really quite striking when you look at karyotypes of cancer cells. You see, this is a Sky karyotyping of 2 cells from a brain tumor, a glioblastoma tumor. And one thing you see is there's barely any normal chromosomes to begin with. And if you look at chromosome 8, for example, the top cell has 4 copies, the bottom cell has 7 copies. And even though the top cell has 4 copies, there's translocation with chromosome 12, 8, 14, and 12, etc. If the authors of the study looked at 25 cells from the same tumor, and actually, this is obviously a kind of a high-level view of the karyotypes, but no 2 cells were alike. And so when I see data like this, and most of you are familiar with the Middleman databases, abound with examples of karyotypic heterogeneity, not just abnormal aneuploidy in chromosome numbers and chromosome structural abnormalities. When I see something like this, the question that comes to mind is the act of being unstable an important driver for cancer evolution? And of course, the obvious answer that is very much true and the subject of many people's work is you generate a lot of heterogeneity, substrates for natural selection, etc. But our lab has kind of, through a series of serendipitous findings, have found additional mechanisms by which the ongoing instability can actually drive cancer progression. And so what does it look like, instability? I mean, again, I'm going to kind of breeze through the introduction, but just for those who aren't as familiar with the topic, when cells divide in mitosis, the phase is called anaphase, and the chromosomes move to the different poles. Normal cells should have nothing in the spindle midzone, whereas cancer cells with high levels of instability obviously have chromosomes that are missegregating. And this is an example of a lagging chromosome attached to microtubules emanating from opposite spindle poles. That's an osteosarcoma cell line in culture. You see chromosome arms kind of squandered around. This is an H&E from a human rectal adenocarcinoma samples, and you see the same phenotype, lagging chromosome. Really nice work done by many groups showed that these lagging chromosomes, if they don't catch up fast enough to the remaining chromosomes, they end up sequestered by themselves in the cytoplasm in structures that we call micronuclei. So these micronuclei basically have very similar double membrane layered structure to the primary nucleus, but it contains usually either a chromosome or a chromosome arm that was, basically too slow to catch up with the, remaining crowd, if you will. These micronuclei are different from the primary nucleus in one key feature, is that the envelope that surrounds those micronuclei tends to rupture very frequently, especially in interphase, exposing double-stranded DNA or the chromatin to the cytoplasm. So this is what a lamin B positive micronucleus looks like, and this is a micronucleus where the membrane and the lamina had just kind of fallen apart. So before I say why do micronuclei rupture, what do they look like in human cancer? And I think this is actually one of the most revealing, IF photos that I've ever seen. This is a human high-grade serous ovarian cancer. And this is a human tumor sample stained with immunofluorescence. And the blue circles are the primary nuclei. The red dots are micronuclei that have ruptured, exposing their DNA to the cytoplasm and recruiting the cytosolic DNA sensor, cGAS. So cGAS is actually stained in red. The reason I really find this picture quite fascinating is, one, this is an advanced cancer. This is not an early tumor. This is an advanced high-grade serous ovarian cancer. There's very few immune cells, and there are more red dots than blue circles. And if you zoom in, you clearly see that these are micronuclei. This is not just schmutz on the slide, if you will. And we've stained over 1,000 samples from breast cancer, melanoma, ovarian cancer, etc. So it is, and another important point is the micronuclei that exist on this slide, the time it was fixed, likely have formed a day or two or three, a couple of cell divisions prior. These micronuclei don't last forever. They either get reincorporated into the primary nucleus as cells divide, they get degraded, etc. And so this tells me that, or tells us that there is an ongoing defect in chromosome segregation, even in these highly aneuploid, highly advanced tumor cells. And this is really important concept here. Why do micronuclei rupture? I'm not going to get into the detail of the mechanism, but there is a lot of work by Martin Hetzer, David Kalman, and others that really nicely discussed some of that work. But recently, our lab as well, along with Stefano Santaguida's lab, have really nicely shown that the reactive oxygen species derived from the mitochondria actually play a double whammy approach. On one hand, they enhance rupture by oxidizing a membrane repair protein called CHIMP7 and changing or mutating its function from a repair complex into a destruction complex, disrupting the lamina by binding LEMD2 and actually breaking the micronuclear envelope. This is all mediated by reactive oxygen species. Whereas Stefano's group has shown nicely that the same reactive oxygen species can oxidize P62, which is an autophagy-associated protein, again, mutating its function, but actually promoting its elimination of the good components of the membrane repair protein complex, CHIMP2 and CHIMP4B, degrading them, and therefore eliminating the chances of repair. So mitochondria seem to be playing an important role, or ROS in general, but we found that it's primarily mitochondrial-derived ROS, in promoting micronuclear membrane rupture and preventing micronuclear membrane repair. Nice work by Martin Hetzer showed that the ER membrane invagination into the micronucleus could also participate in this process. And work by David's group showed that these micronuclei are already defective to begin with. They have lower lamin B2 and lamina components, so they're maybe much more sensitive to any perturbation, even ROS from mitochondria, for example. The rest of my talk, however, I'm going to focus on the consequences of micronuclear rupture. And I can't do it justice in one slide. There's so much work by so many groups here. So certainly but basically I'm trying to kind of categorize the consequences of micronuclear rupture into three broad categories. One, micronuclei have been shown to lead to rapid genomic rearrangements, and I'm sure this has been the subject of many talks in this series. in a single cell division cycle that could lead to chromothripsis and complex rearrangement patterns. Our group and David's group and others have shown that these micronuclei can also lead to histone changes in histone post-translational modifications that lead to epigenetic and transcriptional changes and consequences, some of which can be heritable. And then the majority of the talk, I'm going to talk about the impact of micronuclear rupture on the immune system and inflammation. So first, briefly talk about the epigenetic story, which perhaps some of you have already seen, but just kind of interesting to touch upon. This really started by a talented graduate student in the lab, Albert Agostinos, who was asked a very simple question. What do histone
We don't see your screen anymore.
Oh, sorry. What do histone PTMs...
We don't see your screen anymore, is what I'm trying to say. Something stopped sharing.
Oh.
Yeah.
So where did I lose everyone? Let me try and see.
I could still see the...
Oh, okay. Now,
No.
Yep.
Okay, you saw, I mean, you saw this slide, right? Just, or did I lose you a long time ago? Okay. Can you see now? Just final check.
We can see the screen, Sam. Thank you.
Perfect. Okay. So, so basically, we wanted to understand what are the consequences, the epigenetic consequences of chromosomes being encapsulated in micronuclei. And this first started by a very simple immunofluorescence experiment staining for different histone post-translational modifications in micronuclei and primary nuclei. And Albert actually found consistently in cancer cells, in non-cancer cells, in mouse cells, in human cells, the same changes, where histone acetylation, especially histone 3-tail acetylation, lysine acetylation was reduced in micronuclei, whereas trimethylation in particular was increased, although we also saw loss in ubiquitination, H2 and H2B ubiquitination, K120 and K119. So in order to make sure that this wasn't just like a weird IF phenomenon, Albert went on and did micronuclei purification and did various methods like dot plots for histone ubiquitination, H2 and H2B, and also did histone mass spectrometry in collaboration with Simone Sidoli's group, and found really striking differences between primary nuclei and micronuclei. There's almost kind of a mutually exclusive pattern, as you can see on the heat map here on the right, validating some of the immunofluorescence finding. Some of these changes in histone PTMs happen because micronuclei rupture, and this is very, clear images showing that, this is an example of an intact, two micronuclei that are intact. This is ruptured because we can see cGAS staining. In this case, the H3K27 acetylation is preserved in the intact, lost in the rupture. H3K9 acetylation preserved in the intact, lost in the rupture. Again, the acetylation is often lost upon micronuclear rupture. On the other hand, trimethylation is not lost. And in fact, if anything, seems to be enriched in micronuclei. H2B K120 ubiquitination is lost in all micronuclei pretty much. And other, H2A K119 ubiquitination, which is deposited by this PRC1 complex, is lost in the ruptured micronuclei. So this told us that micronuclei likely have multiple defects leading to different changes in histone PTMs at different stages of their life cycle. And without going into too much detail here, just because in the interest of time, we found that H2BK120 is lost even in lagging chromosomes. So it was born without that mark to begin with, and it was never deposited. H2AK119 ubiquitin and the other acetylation marks are lost upon rupture, and then the trimethyl marks, if anything, are enriched. And we think they are enriched primarily because of the malpositioning of these chromosomes on the mitotic spindle. Some really interesting hypothesis is maybe the Aurora B gradient is actually facilitating the deposition of the histone writers for the trimethyl transferases to be concentrated in the spindle midzone. And this is where the lagging chromosomes are. These features are seen in human tumors. This is again a slide from a high-grade serous ovarian cancer sample, where you can see a primary nucleus and two micronuclei with DNA staining. Obviously, this is human cancer. You can see cGAS very prominently in one of the two micronuclei. This micronucleus is ruptured. The trimethyl mark is preserved. The acetyl mark is lost selectively in the ruptured micronucleus, and this is a quantification. We also went on to look to see if these marks are heritable. We used a very ingenious model that Peter Lee and Don Cleveland have developed, the chromosome Y system. Before the chromosome Y missegregates selectively, which I know many of you are familiar with, but I'm happy to answer questions about the details. But you see that the H3K27 acetylation is preserved where the chromosome Y is, whereas when the chromosome Y is missegregated selectively and then reincorporated the subsequent mitosis, there is an H3K27 hole, if you will. This is where the Y has lost the acetylation in the micronucleus, and then the next mitosis, when it's reincorporated, there is significant reduction in the signal just where the chromosome Y exists. So there could be heritable consequences of these changes. So Albert went on to purify micronuclei, intact rupture, and compare them to primary nuclei and did a TAC-seq on them, also whole genome sequencing. There is no significant changes between the two. That doesn't mean the genome is diploid. This is just normalized to the overall copy number. Basically, there are no differences between micronuclei and primary nuclei. They represent the entire genome. Maybe if you squint, you'll see small differences locus-specific. But here you see significant changes in genome accessibility between micronuclei and primary nuclei, which fits with the fact that they have different histone post-translational modifications. What was really quite intriguing and unexpected is whereas micronuclei overall had a more compact chromatin in line with enrichment in trimethyl marks and loss of acetyl marks, they actually had, wherever they were more accessible, these sites primarily belonged to promoter regions, which was really interesting. And when we did a cut and run to profile histone PTMs and overlay this with a TAC-seq, we found that the promoter regions that were more accessible were more likely to be enriched for H3K4 trimethylation. So I told you trimethylation is enriched in micronuclei. Most trimethyl marks on histone H3 tail are associated with compact chromatin. But in the case of H3K4, trimethylation actually leads to an accessible promoter, which is very interesting and suggests that maybe micronuclei, the changes in histone PTMs are more biochemical. The consequences on gene expression depends on which histone is changed up and down. And there is some follow-on work by really talented postdocs in the lab that is looking at the transcriptional consequences of these changes and whether or not these might serve as a precursor, for instance, for ecDNA formation, increased oncogene expression, etc. So maybe it's not overall indiscriminate transcriptional silencing, maybe it's transcriptional redistribution, taking from everyone and giving to a select few, basically. We then, in order to see if there are long-term consequences for these histone PTM changes in micronuclei, again, we went to the chromosome Y system where you can, and I'm not going to go into the detail of this, it's published, and Peter is actually going to give a talk in a couple of months, so you'll hear it firsthand. But basically, it's a system where we use a DOX-inducible degron to extrude chromosome Y or selectively missegregate chromosome Y. It becomes enriched in micronuclei, and then in the next cell cycle, it can be reincorporated or degraded, etc. By adding a neomycin selection cassette, you're selecting for reincorporated chromosomes, and then you can basically ask, when the chromosome Y goes into micronuclei, when it becomes reincorporated, does it have any long-term consequences on accessibility? And the answer is yes. Actually, we did a TAC-seq on a few clones, either the parental clone and the chromosome Y missegregated clone. And you can see chromosome 6 in all the clones looks very similar between the parental and all the individual clones. Whereas the chromosome Y has a lot of changes. Some peaks or some genomic regions were accessible previously and are no longer accessible. Others were not accessible and became accessible. So clearly there is no reproducible changes in the same locus, but there are kind of changes up and down in different loci, suggesting that what we think is happening perhaps is that the chromosome kind of lost its compass. When it goes into the micronuclei, a lot of these histone PTMs have bookmarking mechanisms to mark where the chromosome was open and closed throughout the cell cycle. And if you have a wholesale change of histone PTMs in a chromosome within the span of 6 hours when it goes to the micronucleus and the micronucleus ruptures, and this chromosome goes back to the primary nucleus, there is a loss of kind of anchoring or bookmarking, etc. So that's where we think is happening. But you can clearly see that not only the TAC-seq is changed, and this is 30 days, by the way, after the micronucleation event and the segregation event. So not only do we see increased variability in chromosome Y accessibility, you see variability in histone PTM deposition as well, and selectively in the chromosome Y and not all the other control autosomes. So this is kind of the model that we came up with based on the data, which is, you have a diploid cell or a normal cell or whatever. When missegregation happens, micronucleation leads to epigenetic changes in the chromosome. The chromosome has a chance of being reincorporated, about 50% or so based on our experience. And then, these chromosomes are altered, and there is persistence, at least for 30 days or so, in the epigenetic landscape or the chromatin landscape of these chromosomes. Another chromosome goes to a micronucleus, comes back, is changed, and this is a way to basically alter the accessibility of the genome without necessarily altering the copy number, and therefore transcriptional heterogeneity. And this was very much the same conclusion that David's group reached when looking at single-cell RNA sequencing of cells with and without micronuclei. Both papers were published together. So the rest of my talk, I'm going to focus on the inflammatory consequences of cGAS, of micronuclear rupture, especially as they pertain to cGAS-STING signaling. So one of the really fascinating aspects of micronuclei is the fact that they expose the cytoplasm during interphase to double-stranded DNA. And when we looked at these early on in 2016 or so, we found that when these micronuclei are ruptured, sorry, are you able to see my screen? I just want to make sure. Yes.
Yes, we can. Yeah, thank you.
Perfect. Awesome. When the micronuclei are ruptured, they become sites of recruitment for cGAS, which is a cyclic GMP-AMP synthase. It's a double-stranded DNA sensor. It is sequence non-specific. cGAS, it's an antiviral enzyme normally. When it recognizes double-stranded DNA, which is supposed to be when a double-stranded DNA virus is infecting the cell, it injects its DNA into the cytoplasm. It leads to the catalysis of a cyclic dinucleotide called cGAMP that then diffuses and binds STING. STING then is an ER-resident protein, recruits TBK1 and leads to its phosphorylation. Leading to IRF3 phosphorylation and interferon gene expression, whereas STING can also lead to NF-kappa B activation. We found that one of the really most striking pieces of data that we observed is that chromosomally unstable cells, if you actually do selective plasma membrane permeabilization, you'll see that their cytoplasm is replete with double-stranded DNA. If you don't permeabilize the primary nucleus, because then it shines really bright and it masks the cytoplasmic signal. And then this is not mitochondrial DNA. If you treat it with double-stranded nuclease, the signal goes away. But really quite fascinating, if you take these cells and fractionate them, make sure there's absolutely no lamin trace in the cytoplasmic fraction, and then subject them both to whole genome sequencing. And Marcin Emilensky actually helped us with this analysis. We were able to recover the entire human genome from a pooled, cytoplasmic fraction of cancer cell lines that had high levels of chromosomal instability, and you can see it here, with no efforts whatsoever. Actually, 5% of the genome in these cells that were highly unstable was present and was able to be recovered from the cytoplasmic fraction. Quite wild, if you think about it. And so what we were interested in understanding is, if cGAS-STING is a pro-inflammatory pathway, how come chromosomal instability is not kind of selected against? How does it not activate the immune system? How do we see it so frequently in cancer, in advanced cancer? And so obviously it can activate the immune system, but how does cancer tolerate it? And this path of inquiry led us to down some really interesting findings, and I'll spend the rest of the talk talking about this. But we found some intrinsic cancer cell adaptations to cGAS-STING that enable cells to suppress interferon signaling, and also cell extrinsic adaptive mechanisms as well. And so some of this project started when we wanted to ask a simple question. We had found previously that chromosomal instability is an important mechanism of cancer metastasis and tumor spread. And we know that there is a cell intrinsic effect to this, because cancer cells that are chromosomally unstable are more mesenchymal, they migrate more, etc. But we wanted to understand if there is an immune contribution as well. So we transplanted cancer cells in 41 and 3 other tumor models that were syngeneic in immune compromised settings and immune competent settings. And in the immune competent settings, we see significant reduction in metastasis, when we suppress chromosomal instability. And this effect was blunted when we transplanted the same cells in severely immune-compromised NSG mice. When we then asked if this could be mediated by cGAS sting, to test this, we knocked out cGAS. And in fact, when we knock out cGAS, we see significant reduction in metastasis. This is bioluminescence imaging of the lungs. Whereas when you transplant the cGAS knockout chromosomally unstable cells in NSG mice, the effect completely goes away. And you see here the bioluminescence. And again, with CT26, wild-type cells are metastatic, cGAS sting knockout are less metastatic. NSG mice, the effect is gone. Another, this is not just like a random CRISPR effect, and we did RNP, ribonuclear protein transfection and not, Cas9 expression, but you see it also, shRNA. You knock out sting, you see significant reduction in metastasis, and this is the H&E of these 4T1s. Is this a rescuable phenotype? The answer is yes. This is 4T1 transplanted in BALC mice. You see sting wild type, you lose sting, you actually lose metastasis. You complement sting at different doses, and this was kind of a serendipitous finding. We used 3 different promoters and actually found dose-dependent rescue of the metastatic phenotype in these cells, which was really quite convincing that in this pathway, likely the inflammatory signaling from sting is an important driver of metastasis. In other words, it gave us a hint that chromosomal instability enables the immune system to collaborate. It enables cell intrinsic signaling to collaborate with the immune system to facilitate cancer progression and metastasis. So to get at the mechanism, or at least get a sense of what's going on, we did single-cell RNA sequencing of isogenic tumors that were either rendered to be chromosomally stable or unstable through kinesin 13 overexpression. I'm not going to go through how this model works because there's many papers on it, but I'm happy to answer any questions. But these cells are otherwise the same until you start changing their instability. Then you have stable cells we call CIN low that are aneuploid, they're heterogeneous, but they are stable. They no longer missegregate as much every time they divide. There are fewer micronuclei, etc. Then there is the unstable cells that are aneuploid, they're heterogeneous, but there's ongoing missegregation. So we're not fixing the aneuploidy, we're not making them diploid again. Transplanted these syngeneic breast tumor cells in the mammary fat pad and actually collected the immune infiltrate and analyzed it with single-cell RNA-seq and recovered really nicely many major classes of immune cells, and found that when we suppress instability in these same cell lines, we see significant increase in the pro-inflammatory signaling like M1 like macrophages, plasma cells, interferon-responsive B cells, activated dendritic cells, plasmacytoid dendritic cells, etc. Whereas these cells, when they're unstable, these tumors are replete with immune suppressive, types of immune cells or microenvironment, as evidenced by M2-like macrophages and myeloid-derived suppressor cells, dysfunctional T cells, etc. So this suggested to us that, well, all else being equal, altering the ongoing missegregation rate in cancer cells likely leads to cancer cell-derived factors that shape the tumor immune microenvironment. And this is a very big question. It can lead us down to a massive rabbit hole. So how do we answer this? So we collaborated with Ashley Lockney's group at Weill Cornell and really talented computational biologists in her lab, Melissa and Ethan, to develop a new method called contact tracing that infers cell-cell interactions from single-cell RNA-seq. And the reason we wanted to do this is we felt that a lot of the methods that were currently in use at the time just gave us too many answers and weren't sufficiently specific to kind of point us to the mechanism. And so contact tracing actually is built on a very simple concept that in a heterogeneous tumor, if there is an interaction between a ligand-producing cell type and a receptor-expressing cell type for that ligand, it's unlikely that 100% of the ligand-producing cells and 100% of the receptor-expressing cells are producing the ligand at the same level and expressing the receptor at the same level and reacting to the ligand at the same level. There is heterogeneity. Some ligands are produced heterogeneously and the receptor expression is heterogeneous. The response to the receptor expression is heterogeneous. And then there is heterogeneity within tumors and across tumor types. So this allowed us to basically stratify tumors based on ligand-producing, ligand non-producing, receptor-expressing cell types, receptor-expressing, non-expressing, and which tumors have both the receptor and the ligand, and which cells have the receptor and the ligands, to understand in an unbiased fashion, and this is really important because many of the other methods rely on teaching, okay, this is what happens when a macrophage sees CCL8-2. But we all know that in vitro and in vivo is very different in the brain and whatever, in the liver, in the lungs. All of these have complex dependence. So we felt that a priori teaching is almost impossible in this method. So we decided to have the method basically exploit the intrinsic heterogeneity in the tumor in order to infer the likelihood of the transcriptional output when you have a receptor and a ligand expression based on this heterogeneity within and across tumor types. And this gave us something, a map that's called the transcriptional response state. And this is conditionally dependent, meaning you compare the transcriptional responses in B cells and macrophages, etc., in NK cells and tumor cells between SYN high and SYN low, or SYN high and SYN high without sting, basically. And so what we found is the biggest changes were seen, for instance, in macrophages, in CCR2 expression macrophages, in the NK cells, in tumor cells. So this told us that the biggest changes happened in these cell types that express these specific receptors. So that was already interesting. We can then map back the individual cells to these transcriptional response states to look at how many cells belong to that state. Is there an increase in the percentage of cells that are CCR2 receptor activated, for example, macrophages, etc. And then we can start directly inferring cell-cell interaction between ligand-producing cells and receptor-expressing cells. This is an example of two cell types, macrophages and tumor cells. And the arrows are directional. So this basically tells you the ligand produced and the receptor expressed. It can go both ways, but just for simplicity. And the blue arrows here indicate the SYN low tumors or SYN low with sting knockdown, whereas the red arrows here indicate SYN high or with active wild-type sting. And you can see, for example, there are ligands that are selectively produced by cancer cells under highly chromosomally unstable conditions, whereas other ones produced by cancer cells under low conditions. All of these interact with macrophages. Different receptors, of course, lead to different downstream transcriptional consequences in these macrophages. Well, we have a nice model. How does it work and how does it compare to other methods? So we compared it to every major cell-cell interaction method that existed at the time. And we saw something really interesting. Did contact tracing actually fit well with other models? The answer is no. It was actually in the outcast group. Sorry for this. In the outcast group here, there's a whole bunch of methods that co-clustered. And then there's three other methods, NicheNet, Connectome, and contact tracing, that each had a separate group. So that told us contact tracing is not aligning with a lot of other models. Turns out a lot of these models here mostly rely simply on the expression of the ligand and the receptor. And as we all know, that mere expression of ligand and receptor is not sufficient to infer an interaction. In fact, many of the receptors are regulated at the post-translational modification level and the protein level, whereas the three models that were separated were ones that relied on downstream signaling. So we felt we should have a better way to benchmark these methods. So we actually used a paired spatial and single-cell RNA-seq data set from human breast cancer samples, and we asked a very simple questions. For non-secreted ligand receptor pairs, if there is a predicted interaction, we should have these two cells co-localized next to each other, because otherwise, how would they interact, especially if the ligand is non-secreted? So it's a very simple question, and we use that because we have the spatial data to benchmark all the methods, and this is the results. I'm not going to go into too much detail, but the bottom line is contact tracing came up with significantly more interactions than all the others, and it ranked them better. In fact, there was a lot of absolute number of false positives, but the majority of the interactions were actually true, by true meaning predicted to have interactions, and then these two cell types are highly localized within the spatial data, and this is the overlap between, and importantly, picked up a lot of the other interactions that were also predicted by the other methods. So what does the method output look like? It can be tabular, etc. But this is kind of an airplane view of the method, in line with my background in kind of chromosomal instability and like affinity for genomics, we thought a circus plot representation, and we'll pay royalty to whoever came up with it, was actually pretty helpful. So the major cell types are on the outside. They are ranked by their transcriptional response state with the key differentiators or diffusion component, pro-inflammatory versus anti-inflammatory, luminal breast cancer cells versus basal-like, T cells, etc. And then the frequency of this transcriptional response state in a given condition. And the conditions here were pairwise chromosomally unstable tumors or CIN low or CIN high with STING knockdown is in blue. So the anti-inflammatory macrophages were enriched in the unstable tumors. The pro-inflammatory were in the stable or in the unstable without STING, suggesting that STING plays a very important role in mediating this phenotype. And then the ligands and the receptors are here. The ligands in bold, the receptors in normal font. Red is the unstable interactions. The blue is the stable interactions. So you can get a big snapshot of the major cell-cell interactions in the tumor microenvironment from a single diagram. And so we then asked, okay, what are the special cancer cell-derived ligands that are produced by tumor cells in response to chromosomal instability and that are rescued when we suppress instability or lose STING? And we found, you know, the top players here in red. We did gene set enrichment analysis of these secreted ligands and found that a lot of known components, EMT, complement, NF-kappa B, but then ER stress kind of was very striking, and it came up in many different analyses that we did. And the reason it was striking is because Nanyang's group showed that constitutively active STING mutations in mice without tumors leads to paradoxical immune suppression. And he actually found that this is because T cells are under chronic ER stress from the constitutively active STING, reducing the threshold for calcium influx into the cytoplasm. So when their T cell receptor gets engaged, you get a whopping dose of cytoplasmic calcium and the T cells basically die. And so that leads to paradoxical immune suppression in these mice. So we asked whether chromosomal instability by ongoing production of cytoplasmic DNA and cGAS activation could phenocopy constitutive active STING. And the answer was yes. So we have STING in these cells. I haven't shown you the Westerns yet, but I'll show you more. And this is kind of a more important concept coming up. We have STING in these cells. When we knock out STING, we don't get any reduction. There's very low basal interferon induction in these cells. These are 41 cells, but they have cGAS, they have STING. If you add cGAMP, there's very little induction of type 1 interferon. But if you add poly-IC, you see a significant increase in interferon-stimulated genes. So it's not that these cells are incapable of producing interferon, they're just incapable of producing interferon in response to STING. But if you knock out STING, you see a reduction in ER stress response. This is cells treated with tunicamycin. With wild type and STING knockout, you see a reduction in most of the ER stress response signaling, BIP, CHOP, phosphorylated PERK, EIF2 alpha, ATF4. So STING is there and is doing something. So we asked if ER stress is important in the immune-dependent metastatic phenotype. And I have to admit that this is probably one of the most striking phenotypes I've seen throughout my career, but you knock out ER stress response mediators and you see significant reduction in metastasis in these triple-negative breast tumors. You put them in immune-compromised settings, there's absolutely no difference. So this might be the downstream mediator of STING in this immune-dependent metastatic phenotype that we observe. So we know that STING is pro-inflammatory. There's a million papers on this. There's no denying that activation of STING leads to type 1 interferon signaling. But in cancer cells, it doesn't seem to lead to type 1 interferon signaling, and it leads to ER stress response. Our hand-wavy explanation for a very long time has been, well, maybe in the acute setting, you get interferon signaling. In the chronic setting, you get reduction in interferon, and maybe STING is downregulated or the signaling downstream of it. But it was never proven in a single model. This is until a talented postdoc in the lab, Emanuele Lettura, decided to have a very reductionist system, IMR90 fibroblasts that are immortalized, they're chromosomally stable, early passage, they have cGAS, they have STING, but they're not active at baseline because there's no instability, there's no cytosolic DNA, etc. And if you add cGAMP on these cells, 6 hours later, you get a whopping interferon response, 100-fold induction of interferon beta-1, CCL5, CXCL10, etc. But if you keep treating them with cGAMP every day, a sublethal dose, they divide, but on day 2, you get 80% of the response. Day 3, you get 50%, 20%, and then 0%. And these cells, after day 6, they have a flat line. They don't produce interferon. But like cancer cells that can produce poly-IC, interferon in response to poly-IC. So it's not that they shut down interferon. It's not epigenetic silencing of the interferon gene locus. It's STING selective suppression of interferon production. But you get the exact opposite with ER stress. At day 5, you get maximal induction of ER stress. At day 1, you had very little. So it's not that you silence STING all of STING signaling. You rewire signaling downstream of STING, where you get reduction in interferon. There's actually a phenomenon called interferon selective tachyphylaxis, or reduced output in response to repeated stimulation, and you get maximal increase in ER stress. And if you treat these cells with a chemical chaperone to reduce further toxic stress, you see a reduction, a flatlining of the ER stress response. So it is truly ER stress induction in these cells. And so we asked, if the pathway is not working, why not just inhibit it? Because it seems to be causing harm, not good. And we actually used the H151, which is a covalent STING inhibitor, Z176, developed by Andrea Blasser, and actually saw a reduction in metastasis in ER stress response in these tumor models that are chromosomally unstable. So this leads us to, I think one of my favorite anecdotes or therapeutic implications of this work. There was an unexpected observation that we made in cells, and this was just, we were knocking cGAS, we were knocking out STING in these cells, and to be efficient, we put the same cells in the same westerns and blotted for cGAS and STING. This is, the 3 cancer cell lines that you see, cGAS levels are low, but functional. You see them in micronuclei. This is what cGAS knockouts looks like, and then this is what cGAS looks like when you knock out STING. On the other hand, when you knock out cGAS, STING levels always went up. It was really quite remarkable. And this is what STING knockout looks like, pure knockout. So this increase in STING protein level when we knocked out cGAS was really kind of remarkable. And what we hypothesized is maybe low levels of STING in cancer cells, while it can be due to reduction in promoter methylation, reduced expression, and all this stuff, it's clearly there, it's clearly doing something. But why does it go up when we knock out cGAS? And really nice work by many groups have shown that STING activation leads to lysosomal autophagy and lysosomal degradation that degrades STING itself. So we asked whether this low level of STING is actually a marker of constitutively active STING rather than underexpressed STING. And because when you activate STING constitutively, it activates lysosomal degradation, and it leads to the degradation of STING itself. So what you're seeing is the steady state levels of STING. You're making some of it, but you're degrading it, and you're constantly at this lower, low steady state levels. When you knock out cGAS, you're not activating STING, but it's being expressed, and it parks at the ER, and its levels go up. But it's primed, perhaps, to be activated. In these fibroblast model, by the 5th treatment, we indeed saw a reduction in STING protein levels. And when we treated cancer cells with baflomycin A1 to prevent lysosomal degradation, we saw an increase in STING protein levels only in the wild type and not in the cGAS knockout. In the cGAS knockout, where STING is not activated, there's no autophagy anyway, or lysosomal degradation, there's no reduction in STING levels, but it's there in the wild type. So that led us to ask a question. Is the tachyphylaxis response that we see in these fibroblasts reversible? And this is the acute induction model, where you see increased induction of interferon-stimulated genes. And this is after 5 days of treatment of cGAMP. The interferon signaling is basically flatlined. If you give these cells a break for 5 to 17 days, they recover again. So that gave us the idea, well, maybe not all hope is lost. Maybe the reason STING does not lead to type 1 interferon is not because it's irreversibly dysfunctional, but perhaps it is just temporarily dysfunctional because it just hasn't had a break. So let's give it a break and see what happens. So this is, let me just go to this. So this is work that was done by a very talented postdoc in the lab, Christy Hong, in collaboration with Benny Zar at Columbia, and was presented previously in meetings, and I'm happy to kind of give you a sneak peek of that story that's currently in revision. But basically, we asked a very simple question. if we allow the system to have a break, and by break we mean either reducing chromosomal instability or knocking out cGAS to prevent the chronic activation of STING, can we then restore type 1 interferon signaling of STING in order to promote, when we activate STING, because now it's parked at the ER and ready to be activated. Of course, you have to give it acutely, otherwise you would mimic the chronic activation. And the answer is yes. I'm not showing you all the data here, but if you actually knock out cancer cell cGAS, you see an induction in STING level, you see an induction in phosphorylated IRF3, but you don't see that in wild-type cells. And the ISG induction is really quite telling. You see a significant induction in ISGs here in the cGAS knockout cells, but not in the wild-type cells. And if you do a double knockout, STING and cGAS, all of that is gone, which means it's truly mediated by STING. Here you give cGAMP or MSA2, which is a STING agonist, acutely, and you see this kind of desired induction that will prime the immune system. Why is this important? It's important because there is a class of drugs called STING agonists that many companies have spent tremendous amount of resources to try and develop based on preclinical murine models that showed very strong effect. But then when tested in the clinic, there was very little efficacy in human tumors. And, I'm not going to go through all, summarize all this data, but this is one of the first STING agonists done by Aduro Therapeutics, ADUS 100. Out of, I think, 47 patients, only one patient had a partial response, and 2 patients had an unconfirmed partial response. Interestingly, in tumors like Merkel cell carcinoma, that is relatively, obviously we didn't analyze these specific tumors, but it's relatively chromosomally stable. And parotid cancers can be chromosomally stable, although not every histology. So back the question whether maybe a lot of these tumors are chromosomally unstable. This is a late-stage patient population with high levels of chromosomal instability. Could there be pre-existing resistance to STING agonists in this patient population, not leading to an adequate response? So what Christy did is said, well, if we take these chromosomally unstable tumors and break the CIN cGAS STING axis by either suppressing CIN or knocking out cGAS, can we restore their sensitivity to STING agonists, which is in this case called MSA2? So we've done this in 7 different models, but these are 2 models here. You see, this is like wild-type tumors with DMSO or MSA2. There's maybe a hint of efficacy, but it's, and these tumors were allowed to grow before we gave the STING agonist. This is an important consideration. Whereas this is cGAS knockout tumors. There's significant, increased response in these tumors. And this is just knockout in the cancer cells. This is a KRAS LKB1 mutant lung cancer model, the transplantable in BLAC6, and this is the 41 model transplantable in BALB/C. And you can see here significant extension and survival. So we perform contact tracing on these tumors to see if there's differences, and there's clearly shifts in the immune microenvironment when cGAS is present or knocked out. But the important control here is that while cGAS knockout tumors are much more sensitive to MSA2, which is the STING agonist, this effect is gone when we knock out STING in cancer cells, which also suggests and supports the hypothesis that this effect is mediated by cancer cell intrinsic STING, which doesn't suggest that the host has no effect. It's just that the cancer cell intrinsic STING signaling, when working properly, is really important in the efficacy of STING agonists. So this is preliminary pharmacological experiment that Christy used, publicly available, cGAS inhibitor RU521, and this is DMSO and MSA2, the STING agonist. The tumors grow relatively unabated, but when you pre-treat these tumors and continue treating with the cGAS inhibitor to give the system a break, and then add MSA2, the STING agonist, when the system has a break, then you get type 1 interferon signaling. The implications of this work is really important because it suggests that we could probably or should probably test the concept of pre-treating patients with a cGAS inhibitor to calm down the inflammation. And then the counterpunch would be an acute treatment with a STING agonist to really allow the tumor to respond and produce type 1 interferon. It's paradoxical because you're inhibiting the pathway in order to activate it, but it's inhibiting the upstream aspects of the pathway to activate the downstream aspect. Neuroscience is full of this. tachyphylaxis response, and it's obviously a very common thing in drug addiction, and the dose needed for drugs to satisfy the exact same excitatory effects or inhibitory effects of neurons, etc. But it's really interesting to see it here in cancer cells in the innate immune system. So we then asked whether cGAS and STING staining could be used as a biomarker to predict, whether the pathway is active or not. And this is prognostic first, where we basically have, we've stained, as I said, close to 1,000 tumor samples. And here you see tumors that are cGAS low and cancer cells STING high. This yin and yang between cGAS and STING, because when cGAS is low, STING is produced but not degraded as much. This is an example of a tumor that is cGAS high but STING low. It's there. You can actually, if you look carefully, you'll see it in the tumor cells, which is very low expression. And we found that tumors that have cGAS low, STING high do much better. This is a cohort with non-metastatic local node at best triple negative breast cancer, and the majority of the metastatic effects happened in the cGAS high STING low cohort. That was really interesting, and the immune infiltrate correlated as expected. So now it would be, we're in the works to try and test, and again, preliminarily, if this method could be used as a biomarker to select patients for STING agonist treatment, because I would expect that these tumors would respond. These tumors would not respond, because these tumors have the pathway, it's ready to act, but it's kind of like at the positions ready to fire. Whereas this one, the pathway is basically overly exhausted. So it has no kind of efficacy. It's like crying wolf, if you will. So this is a summary of the work to date. And I'm going to skip over the last story because it's published, but I think this is kind of an important concept where we feel that chronic activation of cGAS STING in cancer cells, while it might be pro-inflammatory when it first happens, like many other things, as tumors progress, they evolve mechanisms to hide from the immune system, hide from the interferon response that comes out of it. And it leads to this kind of immune suppression. Actually, it's paradoxical immune suppression through ER stress response signaling. Whereas in a normal cell or a cell that gets infected by a virus, there's a robust interferon induction. And of course, as you all know, chronic interferon signaling is not helpful. Even to combat viral infection, our bodies have probably evolved not to exist in a septic state for a very long time. And that's an important, area. But basically, over time, you get an ER stress immune suppressive, microenvironment and in the chronic activation, whereas immune stimulatory in the early. And the question is, can this be used to stratify patients? Can we play a trick, a therapeutic trick, to take patients from this state back to that responsive state, like I just showed you? And I will maybe end with this slide scientifically, just to allow time for questions. This is not the only mechanisms by which cancer cells evade immune activation. In 2021, we published a story about pathway, an ectonucleotidase pathway outside of cancer cells. These enzymes called EMPP1 and NT5E or CD73, which is a very famous enzyme and target of therapeutic efforts. But EMPP1 basically is activated in these chromosomally unstable cells. It's extracellular. If cGAMP happens to leak out of the cells, it can actually be taken up by immune cells and leading to type 1 interferon signaling. So what happens is EMPP1 actually breaks down cGAMP into AMP and GMP, and then hands over AMP to another cousin enzyme, NT5E, that makes adenosine out of AMP. So you basically take an immune stimulatory molecule and don't only degrade it, but turn it into an immune suppressor, which is adenosine. Adenosine can actually promote cancer cell migration and metastasis. It promotes immune suppression. So there is no surprise that the high-grade serous ovarian cancer tumor samples that I've shown you early on that are full of cGAS positive micronuclei, they also have a ton of EMPP1. And there is no T cell around to be found in these tumors. So that basically suggests that when these tumors are chock full of cGAMP or cGAS positive micronuclei, these are probably adenosine-producing factories and actually are used to flip the pathway upside down, turning from an immune suppressive pathway to an immune stimulatory pathway to an immune suppressive one. This is what the EMPP1 looks like. And, this is what the EMPP1 looks like in human tumors. It's really quite striking. It's expressed in cancer cells or in fibroblasts. And wherever we look at from 3 separate cohorts, high ENPP1 expression is associated with poor survival, metastasis, or relapse-free survival, depending on the cohort that we saw. With that, I want to thank many people who did this work, June Lee, Albert, Mercedes, Melody, Emanuele, Christy, and many others and collaborators who helped us, and the funding sources, and happy to take any questions. So thank you so much.
Thank you, Sam. What an amazing talk. That was terrific. So many cool ideas. Thanks for sharing with all of us. So we have a few minutes for questions. Are there any questions in the audience? If so, please raise your hand or write your question in the chat, please. So while people write some questions, I have one, Sam. Well, I have many, but would like to start with. So when you see this epigenetic dysregulation through the translate in micronuclei, have you seen any changes in expression or in chromatin accessibility that might suggest the acquisition of driver events? Is there any recurrence in the patterns, or is it more transcriptional noise?
Yeah, we initially thought it might be transcriptional noise, but this is an area that really talented medical physician scientist Dua Rawi is actually following up on to look to see if there's a rhyme or reason in these, changes. And I think she's on the call here, but I'm sure she'll be happy to, share some of her findings. But this is an area that she's looking at. Until we have more definitive answer, I think the null hypothesis cannot be fully rejected yet, but we think it's not just transcriptional noise.
Okay, thanks, Sam.
Yeah.
So Andrew Jackson, I think you have a few questions. Yeah, thank you.
Hi, Andrew.
Sorry, we cannot hear you, Andrew. Sorry.
Sorry, Andrew. We can hear you.
We cannot, yeah.
Oh, can you hear me or no? Okay.
Andrew, we cannot hear you, I'm afraid. So while Andrew sorts that out, there's a question from Declan in the chat. So Sam, when STING inhibition reduces tumor growth, is it because of increased cancer cell death or immune cell mediated killing, or both?
Why STING agonists reduce tumor growth?
When STING, when you inhibit a STING.
So when we inhibit STING genetically in these models, we don't affect tumor growth, orthotopic primary tumor growth. We don't think that the immune system induced by STING is strong enough, or suppressed by STING in these models, strong enough to actually suppress tumor growth. But in metastasis, we see significant reduction in metastasis. And in the case of STING, we think the majority of it is actually immune dependent. Now, the cells grow perfectly fine in the dish. The primary tumors grow perfectly fine. So we don't think there's a cell intrinsic effect for STING knockout. The STING agonists are, again, the primary mechanism we think is immune dependent, although we haven't formally ruled out this experiment because, well, we did, because when you knock out STING in the cancer cells, there's very little effect on the primary tumor growth. So it is cancer cell STING dependent, and in vitro, obviously, when you have a whopping dose of STING agonists, you can kill cells. So we haven't fully answered that question, but we think it's primarily immune related. It's just that the immune activation signal is arising from cancer cells. But to really formally answer this, we have to do the experiment in like NSG mice.
Okay, thank you. Andrew,
can I have another go at? Can you hear me?
Oh, that works, Andrew. Yeah, thank you.
Okay,
ahead,
okay. So I can repeat the compliment and you can hear it, hopefully, Sam. Lovely talk. I really enjoyed
Thank you so much.
listening. I had two questions. One about thinking about your cytosolic, diffuse cytosolic DNA and its accumulation in SIN positive tumor cells. What do you think its origins are? Have you, how do you think it gets there? How is it generated?
Yeah, that's a really good question. So we did do some exploratory analysis back in 2016 on this. I have to admit we haven't completed it, so I don't have a good answer for you, Andrew, but we know it's genomic, but that doesn't exclude other like DNA sources like mitochondria. The reason being is we actually wanted to be so like conservative by spinning down any membranous structures that I think we even like spun out the mitochondria out of it. I mean, at least it's not, we wanted to just get diffuse nucleic acids. And we found the entire genome from that fraction, even though there was not a trace of lamin in the Western blot fraction that is cytoplasmic. The source, the assumption is that it's micronuclei. When we suppress chromosomal instability, you can quantify the cytosolic DNA content and normalize it to the nuclear. It went down from 5% to about less than 1%. But that doesn't rule out the fact that chromosomal instability, processes induced by chromosomal instability in the primary nucleus would still lead to leakage of DNA into the cytoplasm either. But it is important to mention that this is double-stranded DNA, because I know there are processes that can extrude single-stranded DNA. I think one experiment we did with the chromosome Y is try and actually sequence the cytoplasm for chromosome Y, because that would be a lot more definitive, the system where you kick out chromosome Y into the cytoplasm. And we try, with IF, you can see chromosome Y fragments into the cytoplasm, 3 or 4 days after induction of chromosome Y missegregation, whereas you almost never saw those fragments of other chromosomes. So that's not an exclusion of other mechanisms for why the DNA would go in cytoplasm, but it does suggest that if a chromosome missegregates, there is a likelihood that at least in 5% of cells down the line, the missegregation efficiency is not 100% either, but there is a likelihood that in a large number of cells, you'll see chromosome Y fragments in the cytoplasm, and it's kind of like broken up. This is what we published in 2018. But I don't think we've convincingly sequenced the cytoplasmic fraction with this experiment. It's just technical limitation issues. So I don't have I guess my question suggests that yes, missegregation can lead to cytosolic DNA, but it doesn't exclude other sources.
Okay, thank you. I think that just reassures me that it's still a thorny problem to sort out. But I like the Y chromosome idea. That sounds like an elegant approach to sort it out. My other question was coming from the autoimmune rather than the cancer point of view. I really like your tachyphylaxis experiments. We have a couple of observations which would fit without ourselves, but haven't developed it as beautifully as you have. I guess what I've been wondering is, well, why do we get, for instance, in TREX null individuals or mice, do you still get inflammation? Why don't they just tachyphylax and not inflame? What do you think the difference is there?
Yeah, so that's a really good question, and I've thought about it a lot. And we call it interferonopathy, and there's clearly, obviously, increased interferon signaling in some of these tumors. I don't have a good answer, but I have it's an area of discussion. I just want to share some of the thoughts we had. One is, I think there's a difference between TREX knockout mice and mice harboring constitutively active STING. Because constitutively active STING, every cell is signaling through STING all the time. Whereas in a TREX knockout mice, not every cell has cytosolic DNA all the time and is signaling through STING all the time. So the question we had is, maybe it's a stochastic process. And for whereas for the organism, cytosolic DNA is an ongoing process, but for each cell, cytosolic DNA might be a one-time event that leads to interferon senescence, immune-mediated clearance, and interferon induction in this area. Whereas in that, so it's more of what I think in the interferonopathy, the way the clinical progression looks like, it's not kind of continuous interferon production in every organ and every cell type, in every cell in every organ all the time. It's kind of waxes and wanes. So it makes one wonder whether there might be tachyphylaxis going on. There's outbreaks, and maybe different cells kind of signal at different times. And from a cell's perspective, if you see cytosolic DNA for the first time, even if a cell like 3 centimeters away had seen it like 3 times in a row, last week, it's still the first time unless there's kind of paracrine suppression. So that's, again, this is a testable hypothesis, but, the fibroblast model is actually pretty remarkable. And we've done it in so many different models. And there's actually ongoing data with Kevin Harrington with the phase 1 STING agonist study, a prospective, where we looked at the samples. Again, it's small n, but the data is really quite, surprising given the small number of patients, where cGAS slow STING high patients, pre-treatment, are the ones that tend to, there's a trend towards response. But I mean, I don't have a good answer to really to a really good question, but I think something that's how I would at least approach it.
Okay, thanks, Sam. That's great.
Okay, thanks so much, Sam, for answering all these fantastic questions. So yeah, I think in the interest of time, we've reached the end. So thanks so much, Sam, once again for a fabulous talk, and looking forward to meeting up in person soon. And we'll see you all. Remember, in May, on May 16th, we'll have Peter Lee giving a fantastic talk, so more on why micronucleation and other fantastic aspect of chromosomal instability. So I wish you all a happy weekend. And yeah, thanks again, Sam, and see you soon. Thank you.
Thank you so much. Thank you. Take care.